Yeah, I think it's recording now.
Okay, thank you. This is the Altruism Club, which is a very fascinating term. Thank you for inviting me. Thank you to Sarah Wang for her work on the topic that I will present today. So everyone calls me Dele. I am a professor in the Department of Population Health and Disease Prevention. And I want this to be a good discussion. So I have stopping points in the middle of the slides, just points where we can ask questions. I generally have a lot of slides to present. We don't have to get through them all, but if we go past 5, I hope some of you will stay and that we'll record the whole session so that you can use it to educate maybe others who are not able to attend and ask me questions later on. So effective altruism, it's a great title for a club, and I appreciate what you're doing. So what I want to do today is to talk about the antibiotic resistance, and I'm sure you'll figure out a way that it connects to your goals. So I'll go through a little bit of a background with my work currently, things that keep me busy and kept me up. Been up since 6 a.m., actually 5:45 a.m. today, because there are different parts of the world that need this kind of education. And then we'll start talking about antibiotic resistance as a global challenge. I started working on this with my background in microbiology. That's where my PhD is. So I'll talk about microbial diversity and evolution, sort of the scientific basis of antibiotic resistance. Then I'll talk about gaps in science, gaps in policy, gaps in education, and convergent solutions if we're to curb this problem before it becomes the next pandemic. It already has the implications for that. So right now, I have a major responsibility in the One Health Workforce Next Generation project that's funded by US Agency for International Development, USAID. And there are many partners that collaborate, UC Davis, Africa One Health University Networks, Southeast Asia One Health University Networks, Eco Health Alliance that's been in the news lately for their work in China and the bats and the origin of COVID-19 virus, Atta Health Strategies, Columbia University's ICAP, they are world-renowned for their work on HIV, UC Berkeley, Project ECHO at New Mexico University, and of course, UC Irvine. And my role is to lead the training empowerment segment of One Health, and we'll be talking about the connection with One Health and antibiotic resistance soon. Mostly the goal is that, the health of animals and the health of people and the health of environment are interconnected. So hence the One Health. Environment includes plants of microorganisms and things like that. And antibiotic resistance cuts across all these domains. Our goal is to create a One Health Workforce Academy where we train those who are knowledgeable about topics such as antibiotic resistance and are able to work across these disciplines. So we will launch this website for the Academy sometime this year. Right now it's under development, but it has a lot of content still that can be informative for those who want to explore. As I mentioned, we have a university network in Africa and Southeast Asia, 9 countries in Africa, 7 in Southeast Asia, combined, there are now more than 100 universities and almost 200 departments and schools that are engaged in the network. So some days I'm up at 6 to have a meeting at 6 a.m. with our Africa colleagues, and then at 7 p.m. to talk to our colleagues in Southeast Asia. But the time zone makes it necessary to do that. And that's one of the ways we've coped with COVID-19. So antibiotic resistance, it is a global challenge. I don't know if Sarah has already given you the general talk, but I wanted to let you know that this has become, it's not a recent topic at UCI. Students have always been interested in this topic. And almost 3 years ago now, 2018, a group of students who belong to the Global Health Research Education and Translation, or GREAT Journal Club, invited me to talk about antimicrobial resistance. Was March 12th. It was in person after dinner, talk 7 to 8 p.m. And they run this journal club. I hope they're still active. I know it's been difficult for them, but the work that you're doing on altruism really also dovetails into their motives in bringing speakers to campus, working in global health. So I just want to, some of you may be members of GREAT, that would be wonderful. But I always like to have student clubs kind of collaborate, and I'm sure they can co-sponsor a speaker series with you if you're interested. These were the students at the time. Many of them have graduated now and gone to bigger and better things, but it's a thriving club, and my department supports that club financially. We print t-shirts, we host a journal club, and I'm delighted, if you all in your club want my assistance in any way, I'll be happy to provide whatever support I can. So let's talk about how I got into this field. As I mentioned, I earned my PhD in microbiology, but mostly in environmental microbiology. I was interested in evolutionary processes that drives the different kinds of strains that we see in the microbial world: bacteria, fungi, viruses. And everybody in the world now knows about strains of COVID-19 virus. It's a natural process of diversity in microorganisms. They get mutations, and the mutation is advantageous. The environment selects for them. That's a classic Darwinian procedure. And so I actually wrote a book that was published in 2005 on microbial diversity. If anybody is interested in that book, you can find it online. It really details the theoretical framework for why we have mutations that affect human health and environmental health. And some of them are bacteria, some are bacterial viruses. They cause infection in the lungs. And on this bottom left corner is a bacterium that has magnets in it. Those little red dots are magnets that help the cells align to the Earth's magnetic field. Really remarkable. One of the things I always emphasize when I talk about this book is how inspired I am by those who are really luminaries in their field. And the quote on the front cover of my book was written by Professor Edward Wilson at Harvard. He's really world famous. And he, after reviewing the book, said he wished he had started his own career in microbiology instead of studying ants, although he is well renowned for his work on ants, but because in a pinch of soil, you can have billions of bacteria, all, very wide diversity, and he could just spend his life doing that. And then the preface of the book, or the foreword, was written by somebody I admire tremendously. She's passed away now, Professor Lynn Margulis at UMass. And she was well renowned for the endosymbiotic theory, meaning that, large eukaryotic cells emerge through a combination of, um, you know, bacterial cells entering into symbiotic relationships. And it was controversial at the time, but now we know that the mitochondria that we all carry in our cells are related to bacteria. And she got the National Medal of Science from President Clinton for that. So that they inspired me, and they continue to inspire me to work in the scientific basis of understanding our microbial world. And one of the things I've done is to also write articles about how bacteria evolve traits such as antibiotic resistance. And in this article, published a long time ago now, 1995, I outlined some of the ways that bacteria exchange genes in nature. Some of them, particularly transduction, is by using viruses to move snippets of genetic material from one cell to another. And that's called transduction. Transformation is when pieces of DNA just occur in the environment, either from broken cells, and cells take them up. So if you imagine an antiseptic or soap that kills a bacterium and releases its DNA, that DNA, if it carries antibiotic resistance gene, can still infect another cell that may not have that trait and turn it into a resistant cell. So, and then conjugation is when two cells deliberately come together to exchange genes, very similar to sexual reproduction that other organisms go through. So these three processes are now fundamental ways by which bacteria exchange genes, including genes for antibiotic resistance. You can test this in the lab. So if these little white discs are antibiotic discs, they're embedded with antibiotics. And this is a Petri dish with a sort of a smear of bacterial growth, this sort of brownish slime bacteria. So you can see that this, but on the left side, this bacterium is sensitive to all of the antibiotic discs. So that clear zone around each disc is places where the bacteria cannot grow. Whereas on the right side, different antibiotic discs, some are, the cells are sensitive, but in other cases, there's nothing. They're growing right next to the disc that contains antibiotics. And in another case here, you can see some yellowish clear zone where the bacterium has actually transformed the chemical antibiotic into something else that may be toxic. And then in other places, the resistance is not that strong. And in other places, it's just marginal. So these are easy ways to test whether or not bacteria have resistance. And I've done studies in the field looking at the use of viruses. These are viruses that I personally isolated from lake water. I was working in Tennessee at the time, and it was tedious work. But these bacteria, this bacteriophage, they're called, they're viruses that infect bacteria. You can see them. This green one is a bacterial cell. You can see the phage landing on the cell, and they inject their DNA or RNA, and they commandeer the cell to produce more viruses. And sometimes these viruses carry antibiotic resistance genes, and that way when they infect the cell and inject the gene, the cell can make more antibiotic resistance genes. So that's transduction. And I published this review article in 2008 as a way to show how antibiotic resistance genes are transmitted from one environment to another. One of the things I wanted to do in that paper was to calculate, how many times you can have that transduction event occur in a natural environment to get from one cell that's resistant to antibiotics to having the whole population becoming resistant. And in mathematics, this is a group theory simulation, and I was able to show, this came out of Percy Diaconis at Stanford, trying to figure out, how many times you have to shuffle a deck of cards for it to become completely randomly distributed, that you pick a card, any card will have the information that you seek, or a Rubik's Cube where you, how many times you have to shuffle it to become completely random, right? So it's the same thing. How many times you have to have genetic exchange events in bacteria to have a completely random distribution of the genes? And if you look at and we call it shuffling time, if you look at these numbers, 10 to the power of 11, that's a lot of times. But if you consider that bacteria reproduce very quickly, 20 minutes in a rich environment, they can double, and it's an exponential growth. So it can happen pretty quickly. And that was a good insight into these processes. So just very quickly, like I said, I'm gonna give opportunity for questions, and we can take one or two here if there are any. If not, I'll move on to the next. I can't see you, so I hope the moderator is looking, but you can just unmute your microphone if you have a quick question.
If you're more comfortable, feel free to type it in the chat or
direct message. And then we can also come back to the chat after the whole presentation. So, okay, let's move on then. So how did we come to this problem of antibiotic resistance? Even though we understand it biologically, how it happens, we've relied on it in public health and medicine and agriculture for decades, right? Penicillin was a model antibiotic. It saved a lot of soldiers' lives in the World War II. And, we couldn't have cured the burden of infectious diseases without antibiotics. And this chart sort of, maps out the history. We always knew that there were things in the environment that could kill microorganisms. Salvarsan was invented in 1910, but they're antiseptics. They didn't discriminate. They could damage any kind of cell, including human cells. And so there's a big difference between antiseptics and antibiotics. So in the early 1900s, we knew about antiseptics, chlorine and all of those things will kill cells. But antibiotics, things that you can actually take inside or put on your skin, and they don't do damage to your cell, but they kill pathogens, bacteria. That's a distinction between antibiotics and antiseptics. And that selective toxicity is really critical. And the pharmaceutical industry pays a premium to discover those kinds of cells. So penicillin was kind of the first one. A Nobel Prize was awarded for the discovery in 1945. But as early as 2014, we began to sound alarm that many of these antibiotics, penicillin, methicillin, are no longer working. In 1960, we discovered methicillin-resistant Staphylococcus aureus or MRSA. It causes pimples, but it can also cause flesh-eating disease. So, it's very important that we keep it under control. And so the red zone is really the emergence of antibiotic resistance. And that's going to, if we don't curb it, it's going to reverse all of the progress we've made since 1948. What is the magnitude of the problem? These are, people who have died from antibiotic-resistant infections and expected to continue at least by 2050. 4 million in Africa, 4 million in Asia, almost 500,000 in North America. This was considered the biggest threat in global health until COVID-19. And after we deal with COVID, this will remain a big threat. So it's expected to skyrocket past cancer, diabetes, diarrhea, road traffic accidents. That's why, we're only 30 years away from 2050. And this is a relatively scary scenario if we reverse progress and antimicrobial resistant infections become much more dangerous than cancer or diabetes, which a lot of people fear. So the challenge, of course, is that antibiotics are used not just for human health. We use them for farm animals. Where we don't use enough, then they become resistant and they can spill over from the farm to humans. And a lot of the farm products come to the market. The grocery store may also be opportunities to spread this resistant infection. So we not only have to work with farmers, with physicians in hospitals, but also human education. So many countries are now banning antibiotics in agriculture, in livestock. But the US still a big red zone. We have not done enough. Europe has done quite a bit. In a lot of Africa, it's because they just don't have they don't typically, it's expensive to put antibiotics in feed. But a lot of the effort to get antibiotics out of agriculture remains in the United States. In the US, the CDC, more than 23,000 people die every year from incurable infections. 250,000 fall sick, and prolonged sickness that's not curable can do long, even when somebody survives, it's not going to be pretty because they'll have a long recovery period. And one particular pathogen, Clostridium difficile, kills 14,000 people every year. That's a major challenge for the healthcare industry. And people find it in microscopes, used in hospitals, and it's really very difficult to get rid of, hence the name difficile, because it forms spores. Questions? This is a time to type questions in the box or ask. So I'll just pause for a couple of minutes.
There are a couple of questions in the chat. Yeah, the first one from Brenda. She says, hello, Dr. O, what are the best ways that we as students can make a contribution to reduce antibiotic resistance?
Good question. It's education, and I will get to that in another section. So you have to educate yourself, especially what you buy in the grocery store and when you go to the hospital if you're sick, you have a flu, to know that antibiotics work only for bacteria and fungi, not on viruses. So you can't put pressure on the doctor or the nurse practitioner to prescribe antibiotics because it won't, they have to do proper diagnosis. You saw those that Petri dish plate and the antibiotic discs, that's the kind of test they do in the hospital to know which antibiotic is right for you. Okay.
All right. Yeah. And then Chrysalin asks, hello, thank you for your presentation. I was wondering if the effects climate change will have affect antibiotic resistance issues, as I have heard many pharmaceuticals are isolated from plants and fungi. With more deforestation occurring, how will this affect our antibiotic resistance issue?
Yeah, it's a good question. It's a question for research at this point. One of the subjects of One Health is the impact of climate change on things like antibiotic resistance. You know, one of the research that I've done, for example, if you just take a look at global warming, if the temperature increases, bacteria tend to grow more quickly. That's why we have some certain kinds of infections increasing in the summer, especially for bacteria, because under hot conditions, they're dividing more rapidly, mutations are accumulating. But you're right, deforestation also brings us more close to wildlife. And to the extent that bacteria that may be endemic in wildlife, but new to the human population, may introduce genes that carry antibiotic resistance that then make us very vulnerable. So that's one of the biggest threats. It's how human populations are moving closer to wild populations and wild type pathogens that may cause damage. But we still need a lot of research to map out those hot spots where those kinds of events are vulnerable to climate change.
Okay,
Good
so a lot of
questions.
research.
Yeah.
Okay, and then the next question is from Crystal, and she asks, what are some alternatives for antibiotic medications?
Yes, we will talk about that as well. In a lot of flu infections like sore throat, one talks about rest and drink water, but generally it's about getting vaccinated for many of the pathogens, bacterial pathogens that we have vaccines for, like tuberculosis, right? So that's the best prevention. And then anything you can do to boost your immune system to resist infections, good diet, nutrition, those are really great preventive methods that can counter antibiotic resistant infections. And the same things we do with the regular hygiene, washing hands, not touching your face all the time, don't pick food off the floor. Those are really great ways. Okay, let's move on and we'll save more questions for this section. So what are the gaps in science, right? So one of the things I've done, as I mentioned, we have an understanding of diversity and evolution. This article was published in the Encyclopedia of Evolutionary Biology. But there's nothing we can do to prevent pathogens like Staph aureus or tuberculosis from becoming resistant to antibiotics, right? That's the way they survive. It's a race between us and them. So the more new antibiotics we invent, the more likely they're going to develop resistance. And so you can begin with, just a wide collection of cells with 2 of them resistant to antibiotics. When you use antibiotics, you kill off all the blue ones that are not resistant to antibiotics, right? And then the antibiotic resistant ones become dominant in sphere 3 here. And then they can transfer that gene to other non-resistant infections. So ultimately, we're going to end up with a world where almost every organism is resistant to antibiotics. So we need to break it at this point between 1 and 2, so that when we use antibiotics at 2, they're extremely sparingly, not excess, and only ones that actually work that we should use, and to test whether or not they're resistant cells in the population before we use it. So that when you use an antibiotic here, it's a broad spectrum one that kills everything, not the two that are already resistant that then can become a big problem. So again, meat products can spread it, contaminated water from farms, and people sneezing and coughing without covering their mouth, and wastewater from healthcare facilities as hospitals are all opportunities for spreading. And so education targeting each of these areas is really important. At the international level, there is a global action plan that the World Health Organization has been nurturing for antimicrobial resistance. It's called GLASS, the Global Action Plan for surveillance. So we have monitoring stations in every country, making sure that we capture antibiotic resistance. One of the conferences I attended in 2017 when I was working in the US Department of State, which has a really strong interest in this topic, was an environmental dimension of antibiotic resistance, because we know little outside the hospital and the farm. And so recently, the UN Environment Program and the World Health Organization have agreed to collaborate on topics such as antibiotic resistance. So it's not just the human health sector, but the environmental science sector that can move us forward. And when I worked in the US Department of State, the Office of International Health and Biodefense, we collaborated with the Society for Environmental Toxicology and Chemistry and the US Geological Survey to undertake a study mapping the life cycle of antibiotics as they go from pharmaceutical factories where they are manufactured, but their wastewater may be deposited, to disposal of antibiotics from homes, flushing antibiotics in the toilet when they expire is a big problem, to farms, to people, and to storage. So we were very interested in creating the methodology to bridge the science gaps in this cycle. And we held a workshop, my colleagues in the office in Singapore, to bring countries together, 12 countries, to really understand. So for example, if you buy fish in Albertsons or shrimp, very likely some of that comes from the lower Mekong River system in Asia. And we need to educate people there about the use of antibiotics in fisheries. Otherwise, their product will not be sellable in the US. And even if it sells, we may be importing antibiotic resistance from such foods. So I had many outcomes for the workshop that we ultimately presented to the countries. And Jessica Petrillo was my collaborator in turning that workshop outcomes into a publication that you can download on the web. It was published in 2018 in the journal Environment International, Emerging Issues in the Environmental Context of Antibiotic Resistance, because we saw it as a major scientific gap in our understanding of how antibiotic resistance spreads in the environment. So we listed 4 opportunities for intervening under the supply chain: manufacturers, distributors, storage, and counterfeit antibiotics, which are common in Africa and Southeast and Asia, and maybe even South America. And you can find some in the US as well. And they are usually low dose, so they can encourage resistance to develop. And then in usage, how we dispose of expired antibiotics, wastewater treatment, solid waste collection, compliance with treatment course. So when you're prescribed with antibiotics, to make sure that you take the full course. And then in waste management, industrial effluent, domestic sewage, landfills, and recycling processes. So there are major gaps in understanding the evolution of antibiotics, the environmental context, because we don't sample enough, but that workshop by US Geological Survey, the Society for Environmental Toxicology and Chemistry, and the US Department of State really are framing those questions so that the research community can address them, and then we can use the results to do intervention. So in the interest of time, I'm going to keep going, but I use this opportunity to type in questions so that you don't miss out. So I want to talk about gaps in policy. So President Obama, before he left office, actually issued an executive order combating antibiotic-resistant bacteria. It was issued in 2014. It was a big deal because he saw it that this is a big threat to US healthcare system. And you can still find the text of that executive order if you search the archives of the White House. So the executive order required that all of these departments, and you may wonder why, Department of State, of course, because we do health outside the US, but the Department of Justice, we talked about counterfeit medication, Veterans Affairs, Homeland Security, all of these agencies are involved in antibiotic resistance policy and enforcement. So if you're interested in policy, you will see that, in the US government, this topic is not a simple one. It's multidisciplinary and multi-sectoral. And the president, one of the requirements is that we have an advisory council on combating antibiotic-resistant bacteria. When I worked in DC, I attended one of their sessions and gave testimony about the scientific need to understand better the environmental context. PACAB still meets regularly, but again, it suffered a little bit under President Trump's term. I think it will come back now as an effective way to deal with the policy issues around antibiotics under the Biden administration. The other aspect of policy is to work with pharmaceutical industries about their role in pushing antibiotic sales to hospitals and to doctors and nurses. Sometimes they want to just sell, but they oversell, and then they flood the market, and then resistance develops. They also don't invest a lot in developing new antibiotics because it's expensive. Well, they need to do that, and then they need to voluntarily monitor their wastewater so that it doesn't contaminate rivers and landfills. So the United Nations issued a high-level political declaration on antimicrobial resistance. It's the first one on a topic like this ever. This was 2016, really putting that policy up beyond national policy to international policy. And this is a major opportunity. If a student is interested in studying international governance on antimicrobial resistance, this is a great place to start. It was considered a major success when this declaration was approved by the General Assembly. So it shows you how important this topic is to global health. So let me now talk about the work of some of my own students and collaborators. So on the policy gaps, Dr. Tamara Jima was a PhD student who worked with me. She got her PhD in public health, the concentration in global health. Now she's a postdoctoral researcher at UCI School of Nursing, and we're still working together. So she's actually half Russian, half Ghanaian. So she was interested in understanding how the Ghana national policy on antibiotics and antibiotic resistance is being implemented. So She went to Ghana, she trained a lot of people and did a survey of knowledge, attitudes, and took really great pictures. So if you're interested in global health, she's a good example of how busy you get and the opportunities you have. So these women were in focus groups that she assembled, and they show off the information document that she distributed. So we've published several articles, and many of these are available to you online. But if anybody wants copies, I can also share them with you. The first one was published in the journal One Health Outlook. And this is the study of knowledge, attitudes, and practices among different communities in Ghana. So she looked at the rural and the urban populations, agriculture use, hospital use. And in Ghana, apparently there are some street level pharmacy shops, chemists that you can buy antibiotics, just like in the US, you can buy antibiotic ointments at Albertson. In England, you cannot do that. So there are these really policy differences that are very interesting to study. The other paper that we published, social demographic characteristics. So, the level of knowledge that people have affects their what we call stewardship of antibiotics, what they think of therapy, how they use it, do they comply with the doctor's recommendations. So this was published in the Journal of Global Health Reports, and we published another one on the policy of Ghana, the National Action Plan, stakeholder analysis of how that plan is being implemented. Really excellent papers that Tamara worked on. So in terms of policy gaps and the work with Tamara, you may have questions as well. I will reserve the time for those, but use this time to write them down. I want to talk about gaps in education. And this is the work of Sarah. I didn't tell her I would talk about her work, but she's, as you all know her, she's an honors research student. And her honors project, she's very passionate about this topic. So, she, since last year, she's been working on the knowledge that UCI students have on knowledge of antibiotics, their attitudes and practices. And this paper we're working now, I'm doing the final revision, will be submitted to the Journal of Adolescent Health, in part because adolescents represent that transition point between going to the hospital by yourself when you used to go with your parents, or shopping for groceries independently, not eating at home. So how you know about what to do and what not to do regarding antibiotics is critical. So Sarah did a lot of work here. 211 individuals participated in a survey that she developed based on the World Health Organization's survey instrument. And she learned a lot of statistical methods to analyze them. And one of the findings, and she may already have given you all the same survey, but one of the findings that junior students significantly know less about antibiotics than seniors and sophomore. Sarah will correct me if I interpret the data wrong, but I'm sure I'm right. And so there is an opportunity here to introduce a course that's available, like the freshman seminar for all students. We couldn't do freshmen easily because they weren't, this was done in the summer and they were not here yet. But clearly, I think it's important to have a course to increase the knowledge of antibiotics among students. And then there are differences in gender. I think females tend to know more because they're used to paying more attention perhaps to health issues than men. So we can target these educational opportunities accordingly. So this is a major finding, not published yet, but I hope that it will be published soon. The other work that Sarah is doing is to create a storyline to improve the knowledge. And this is an excellent project that I'm sure she can talk to you more if you invite her to give a presentation on her honors research project. And in a few days or so, she will give a talk to the invitation by the Food and Drug Administration on antibiotic-resistant gonorrhea. She worked on these slides in the last couple of days. And your age group is very vulnerable to gonorrhea. It's one of those growing venereal diseases, sexually transmitted diseases, particularly females aged 15 to 19 and 20 to 24, and males 20 to 24. You know, it's just 4 years ahead of the females. So when you cannot cure gonorrhea, it's a big problem for the US and for the world because of the sequelae in ectopic pregnancy, infertility, and the risk of getting HIV is higher. So we've seen antibiotic resistance emerge. All of the antibiotics that we used to use to treat gonorrhea, penicillin, ciprofloxacin, and cefixime have become resistant as of 2014. And the only, the last resort antibiotic right now is ceftriaxone, which the CDC recommends 500 milligrams, a single therapy. And if you get resistance to that, that will be a big problem for the world. So Sarah has engaged many students, some of you may be there, to really think about early education, creating a curriculum for the K through 12, and working in California school districts with lower socioeconomic status. So that's a very important work that she's doing, and I applaud her for doing that. So you may have questions about this as well. I want to get through this last section so that we can answer more questions, but just giving you a pause here to write down your questions. So what are the solutions that converge? In 2018, I attended the Prince Mahidol Award Conference in Thailand, in Bangkok. And there's a lot of interest in making the world safe from the threats of emerging infectious diseases. Of course, in 2018, we wish we'd known about COVID-19, but it shows you that we cannot really predict the future, but we must be prepared, right? So I attended at PMAC the session on antimicrobial resistance. And some of the things we were talking about at the time, I think COVID has pushed it to the back burner, but this is a big threat that we cannot afford to take our eyes off the ball. And I'm looking forward to, at the time we had the new director of the World Health Organization, Tedros, give us a talk on WHO's fight on antimicrobial resistance. One of the things I'm happy about is since that conference, we've brought in, I did point out that we need the United Nations Environment Program to become part of this fight, and that has happened. So just when I was in Thailand, we visited a farm that's practicing the best kind of prevention against antibiotic resistance. They don't use antibiotics in their livestock, but they also worry about people bringing in antibiotics into the farm. So we have this, you had to enter the farm, you had to walk in an antiseptic solution so that whatever soil you're bringing in is washed off and disinfected. And in the farm, this, you can imagine a farm that doesn't practice antibiotic resistance. When you have pigs wallowing in water, I took this picture just to show how they make sure that their wastewater is not dispersed. It's treated before it goes into groundwater and then is filtered. And you have antiseptic soap all over the place to make sure that the germs do not leave the farm area. So that's best practice. What I learned from working across these countries, just to think about convergent solutions. And this is a new development. And I'll talk about another graduate student who's working with me on this topic, Gabby Gossin. And so she's pursuing a PhD in public health with a focus on disease prevention. And just yesterday, we got word that we were one of the awardees for this is a public health global grant, which is a global network for academic public health for Latin America, for Asia Pacific, for Africa, for Europe, for the United States, and for Southeast Asia. So these groups came together and we submitted a proposal that is titled Antibiotic Stewardship is Public Health. And here's a little abstract. So Gabby will be the global ambassador and I will be the PI. I'm going to invite Sarah to work with us because she is also very passionate, like Gabby is, about this topic. But also any one of you that's interested in the global context of antibiotic resistance, I'm inviting you to come work with us. So I just want to conclude, we have a big problem that is solvable in antibiotic resistance. It's very urgent. It's a big challenge for global health, but it's not insurmountable. We can see it coming, unlike COVID-19 that we saw it coming, but we really didn't know what to do. Here, we kind of know what to do and we can prevent it. It's a big One Health challenge because we have to work with people, with animals, and the environment. And the solutions will require that we bridge gaps in science and policy and changing behavior through education. And finding those gaps in education, just like what Sarah's project has been doing. So here's an opportunity for questions. Before we get to the questions, if some people have to leave at exactly at 5, I want to invite you all to the UC Global Health Day, April 24th, that's next Saturday. We are 10 campus strong, and we talk about these challenges in global health, including antibiotic resistance. It's a virtual conference, there's no cost, but you have to register to get the Zoom link. So UCGHI, if you just type that into Zoom, you'll find the link. Register and attend. The focus is decolonizing global health to advance health equity, but there will be all kinds of opportunities to learn. And you can, of course, ask me questions after today. Just send me an email or talk to Sarah. She knows how to reach me. I try to respond very quickly, but please share this knowledge that you received today with your colleagues so we can curb this problem from becoming a pandemic. So thank you very much. Questions? I think we still have a moderator who can call out the questions.
Yeah, so we still have, Vanessa asked earlier, why do you think heart disease is much more recognized as a threat to humanity rather than antibiotic resistance?
Excellent question. Before COVID-19, even in my family, because I'm a microbiologist, I have seen viruses under the microscope. I have firsthand knowledge of what they are. 99.9999% of people in the world, it's an abstract concept. They have never seen, bacterial cells. They haven't seen viruses. Right? So convincing people, 6 feet distance, washing hands remains an abstract concept. But a lot of us, maybe not everybody, but when somebody has a heart attack, it's covered in the news. We can actually see the person fall down or in the hospital. So I think things that seem more visceral, more close to us. Somebody clutches their chest, and then we fear them more than things that we can't see, right? So I think COVID has changed that a little bit. People are now kind of aware of, that you have to sort of maintain this sterile bubble around you, that the air we breathe in and breathe out is shared with other people, and that air may contain pathogens. But still, it's a little bit distant. But there's also an American culture part of this. If you grow up in places where clean water through chlorination is not guaranteed, where sewage treatment and toilets, pit latrines, you're more aware of the dangers of infectious diseases, of mosquitoes, of diarrhea and cholera, than if you live in the United States and you take it for granted that your water is clean, you flush the toilet, it goes somewhere you don't really understand, but it's gone, right? So I think there's that kind of knowledge. We shouldn't assume that developing countries have a poorer knowledge. They actually have more awareness of these risks than in countries where we've taken it for granted because we have plumbing. So I think that's why.
Awesome, thank you. Ishrat asks, do you think that the 2050 death expectancy might approach earlier due to overprescription of antibiotics during COVID?
Yeah, I don't know. The data is not out yet about what has been prescribed in COVID. I think it was pretty clear that antibiotics won't cure COVID, despite what our President Trump was saying about other kinds of drugs, like the anti-malarial drug. There was a lot of confusion about what would be a cure for COVID, but antibiotics were never seen as a cure. I think maybe in Brazil there was some discussion, and our president talked about antiseptic. Remember, he was asking Burks, what if people inject themselves with or drink antiseptics, right? That was bad misinformation. But we may see ultimately that people are more aware and demand more antibiotics as a result of just getting more knowledge of health and infectious disease. I think that data needs to be collected. I don't know that life expectancy in the US has declined in 2020 just because of COVID infections and deaths. But I think we underestimate the decline, because when we tally up everything else that killed people during that year, it's a lot more. But I'm not sure antibiotic resistant infections would have gone up. First of all, most of us avoided such infections because we stayed away from other people and stayed home mostly. So if anything, my guess is it probably was reduced, the number of infections that need antibiotics, and therefore antibiotic resistant infections. But we didn't break the cycle.
All right, and then Brenda asked, are there any open opportunities in research or policy for students? Are there any opportunities with you at the moment?
Well, right now, Sarah can tell you, I work with Sarah on our honors research. She's a public policy student, and she has 3 or 4 other students that I'm supervising in 198, but she's really working with them. Sarah, if you have openings, let the group know. But because of my One Health project with USAID, it's really difficult for me to take on additional students because I want to do a good job, and but I just encourage people to email me and we can talk and check. There are some issues with the global policy, the United Nations policy, that I think needs more people to work on. There is the This is Public Health Global project that we just started that will require work in Ghana and Senegal. So if somebody speaks French, there's an opportunity there. But yeah, send me an email and we can discuss. But very few opportunities now just because of being too busy. Yeah.
I've been in contact with Elaine, who is the director of the Michigan Antibiotic Resistance Reduction Coalition, and she is offering opportunities to expand antibiotic stewardship in our community and potentially do research to present at
Excellent.
conferences or present.
Good.
So
that's excellent. Yeah. So please contact Sarah. Other questions?
Were there any other questions that I missed in the chat?
Oh, I'm impressed. We finished close to 5. I had a lot of slides. I hope I didn't go too quickly, but I really excited about the opportunity to share this with you. Lots of students have contributed to the work, and in addition to just learning about the work, I encourage you to also spread the knowledge.
We're really excited to have you here. We really appreciate you coming and then answering all of our questions as well.
Excellent. Well, enjoy the weekend and be safe, get vaccinated, and keep up the good work.
Thank you, Professor.
Thank
All right,
you.
thank you.
thank you.
Thank
Take care.
you.
Bye. And when you have a link to this recording, you could share it with me. I'll appreciate it. And I welcome questions from those who watch it later. Okay. Bye, everyone.
Oladele.
Goodbye.
Bye, Sarah.